Dear students, welcome to EPG Pathshala. I am Dr. Y.S. Rajput, Emeritus Scientist, Animal Biochemistry Division, National Dairy Research Institute, located at Karnal in Haryana. Today, I am going to teach you about the southern blotting technique. With the discovery of agarose-gel electrophoresis, it was possible to separate nucleic acid, which includes DNA and RNA, based on their molecular size. Electrophoretic techniques are very handy for analysis of charged particles. However, the method lacks high degree of resolution when sample is complex and contains molecules similar to analyte. When other scientific principles coupled to electrophoretic resolution techniques are added, then degree of resolution is enhanced. This is the concept in blotting techniques, where molecules separated on gel are transferred to for their subsequent analysis. Edwin Southern in 1975 transferred DNA fragments separated by electrophoresis to a membrane, and the molecules of interest was detected by some probe which was hybridized with the membrane. The concept gave the basis for the range of techniques for understanding the organization and expression of genetic materials, and was named as Southern hybridization. Subsequently, procedure for RNA was developed, which was referred as Northern blot, and for protein, which was referred as Western blot. In this lecture, we will elaborate Southern blot technique. The focus will be on how DNA can be transferred from agarose gel to membrane. The objective of presentation is to understand the basic concept of Southern blotting, to make you understand practical aspect of Southern blotting, and added objective is to explain the applications of Southern blotting. Blotting is a technique used for transfer of DNA fragments, RNA, or protein separated by electrophoresis on gel to a rigid membrane system, and subsequent detection of these molecules by using appropriate detection method. The Southern blot is used for DNA, Northern blot for RNA, and Western blot for protein. Blotting was developed initially for DNA by Sir Edwin Southern, and the technique was named after its inventor as Southern blotting. Further, other blotting techniques developed for RNA or protein developed on the same principle were named conventionally analog of it as Northern blot for RNA and Western blot for protein. Southern blotting can be described as a procedure for locating a desired nucleotide sequence or gene from a mixture of DNA molecule. The fundamental principle of nucleic acid blotting relies on the principle of filter hybridization. It essentially involves 3 steps. In the first step, different fragments of DNA or RNA molecules present in crude mixture are separated according to their size in agarose gel electrophoresis. Next, DNA or RNA fragment separated on agarose gel are transferred to membrane. This transfer is achieved with the help of capillary action. Finally, DNA probes are often used to detect the nucleic acid molecules with sequences that are complementary to all or part of the probe. The probes are complementary nucleotide sequences to all or part of the nucleic acid and are labeled and are used to detect nucleic acid molecules. If the probe finds its complementary site in any DNA or RNA molecule, it will bind to the site and will provide a specific signal after autoradiography. If molecules of specific size are detected with the probe, we can be confident that hybridization experiment was specific, and we can find out the location or presence of desired gene in fragmented DNA molecules. The specificity of experiment depends on the washing of the probes after binding to the complementary site. If at this step the extra probes are not removed from the system, it will give false signal leading to a wrong interpretation. The probes used for hybridization are so sensitive and selective to the nucleotide sequence that as low as one molecule of the sequence present per cell can be detected. Thus, it is also possible to determine the number of copies of a particular sequence of DNA present within the cell. If the nucleic acid hybridization reaction single strands DNA or RNA randomly collide with each other in a number of all possible alignments, under the stringent condition, only one perfectly matching molecule will hybridize. The basic steps in nucleic acid blotting starts with the electroforces of nucleic acid, transfer of separated nucleic acid from gel to membrane, immobilization of transferred nucleic acid in membrane, pre-hybridization, hybridization with probe, washing of membrane for removal of unbound probe, and detection of signal. It is made clear that Southern blotting is a multi-step technique. Thus, it is essential that each step in Southern blotting is performed carefully. Also, probe should be specific to allow correct interpretation of results from Southern blotting. In remaining part of my presentation, the focus will be on practical aspect of Southern blotting technique. This will include preparation of DNA fragments, electroforces of prepared DNA, blot transfer of DNA fragments from gel to membrane, hybridization of membrane with a specific labeled probe, and analysis of results in autoradiogram. The initial requirement of Southern blotting is the preparation of DNA samples and their fragments. The DNA sample should be purified and is available in adequate amount to perform the experiment. During this step, the DNA sample to be analyzed is fragmented into small fragments which can migrate through the matrix of electroforces gel. Restriction enzymes are used for cleavage of genomic DNA at specific sites to generate small fragments. Many times, a combination of restriction enzymes is used. This preparation gives a set of DNA fragments which can be easily separated by gel electroforces. For selection of restriction enzymes to be used, it is required to know the restriction sites present nearby the gene of interest. A specific gene can contain a number of restriction sites, and cleavage of the gene with particular enzyme produces fixed number of fragments of the fixed size. Mutation, deletion, and rearrangement is a particular gene of interest may alter number or the size of restriction fragment generated from enzymatic cleavage. This type of change in restriction pattern is called restriction fragment length polymorphism, or popularly referred as RFLP. After fragmentation, DNA sample is concentrated by using phenol chloroform extraction and ethanol precipitation for performing the next step in electrophoresis. After digestion of DNA with restriction enzyme, the digested DNA fragments are separated according to their molecular size by means of technique called agar-gel electrophoresis. In agar-gel electrophoresis, separation is based on size of nucleic acids. Nucleic acids do not require any treatment for making their charge to mass ratio constant. Nucleic acids have negative charge, and amount of net negative charge is proportional to size of nucleic acid. So, charge to mass ratio of nucleic acid is constant. Large size nucleic acids move slowly, whereas small size nucleic acids move faster in agar-gel electrophoresis. In both agar-gel electrophoresis as well as HDS space, separation is based on size of nucleic acids and protein respectively. In both cases, charge to mass ratio of respective macromolecules is constant. In HDS space, this is achieved by binding of HDS to protein, which acquires net negative charge. The binding of HDS to protein is almost constant for wide range of protein, and for every 2 residues of amino acids in protein, 1 molecule of HDS binds. HDS binds to hydrophobic pockets in protein, and in doing so, sulfate groups are exposed out. Nucleic acids do not require any treatment for making their charge to mass ratio constant. Nucleic acids have negative charge, and amount of net negative charge is proportional to size of nucleic acid. So, charge to mass ratio of nucleic acid is constant. Large size nucleic acids move slowly, whereas the small size nucleic acids move faster in agar-gel electrophoresis. Size of nucleic acid and size of pore in agar-gel determine the magnitude of frictional force exerted on nucleic acid in their movement. This situation is similar to HDS space. Pore size of agar-gel is dependent on concentration of agarose. Size of nucleic acid should be such that these are able to enter pores of gel, and all components in mixture should experience variable frictional force to allow their separation. Appropriate concentration of agarose is used for preparation of agarose gel. This is dependent on number of base pair present in digested nucleic acid. For example, 0.5% agarose is used for fractionating 10,000 to 15,000 base pair long DNA. Similarly, 2% agarose is used for fractionating or separating 100 to 2000 base pair long DNA. So this range is given in tabular form here. The diagram before you gives overall view of arrangement for transfer of nucleic acid from agarose gel to membrane. As you may observe that arrangement involves placing gel and membrane in close vicinity, and these two are in contact with Wartman paper for easy flow of solvent from gel to membrane through capillary action. The most common arrangement of southern blotting uses capillary action in the direction of flow of molecules from buffer to membrane. The assembly can be easily prepared in the laboratory. Although electrophoretic transfer of nucleic acid from gel to membrane is feasible, as in case of Western blot, the assembly as shown in diagram is equally effective in transfer of nucleic acid to membrane. The assembly essentially has platform for placing Wartman paper, gel, membrane, adsorption paper, and weight in sequence. Since solvent flows upward through capillary action, all portions of Wartman paper, gel, and membrane must be kept wet, and air bubbles should not be trapped while layering over each other. For success of transfer, Wartman paper larger in size than that of the gel is cut and immersed in buffer and then placed over the support. Only edges of Wartman paper should dip in buffer. The gel is placed upside down on Wartman paper. Dear students, so far we have covered principle of blotting techniques in general and southern blotting in particular. Southern blotting gave birth to other blotting techniques. In contrast to Western blotting, southern blotting can be performed without electrophoretic transfer. Capillary flow of buffer from Wartman paper to agar gel and from agar gel to membrane carries nucleic acids from large pores of agar gel to membrane. No specific equipment is required. The device for transfer of nucleic acid from gel to membrane can be easily prepared in the lab. Detection of nucleic acid on membrane is achieved with the use of DNA probe, which has complementary nucleotide sequences to target DNA. Specificity of probe is important for detection of target DNA, and the equally important is how to avoid non-specific reaction. In next few slides, I will be explaining more details of how the southern blotting is performed and what are its applications. The steps followed for southern blotting are described in figure before you. In the initial step, DNA sample to be analyzed is purified and fragmented using restriction enzymes. In next step, the fragmented DNA is run on agar gel electrophoresis, and now the DNA fragments are separated according to their molecular size. After separation of DNA fragments in gel, the fragments are transferred to a solid support, mostly the nylon or nitrocellulose membrane, using the assembly as described earlier. After blot transfer, the membrane is initially baked in oven, or it can be exposed to UV radiation. In this step, the DNA transfer to the membrane is permanently attached to the membrane. In next step, the membrane is treated with a specific DNA probe, and the probe is hybridized with specific target DNA fragments transferred on the membrane. In next step, after hybridization, is to visualize the hybridization pattern on X-ray film by autoradiography. If any chromogenic probe is used, then color will be developed on the nitrocellulose membrane. DNA probes are single DNA fragment with a specific sequence whose presence in target DNA is to be determined. The probe is labeled with a radioactive, fluorescent or chromogenic tagging. In many cases, RNA can be designed in place of DNA as a probe for ensuring the specificity of probe to the sample DNA sequence during hybridization. Salmon sperm DNA is used for blocking the surface of membrane. After hybridization is over, the excess of the probe is washed off from membrane by using saline sodium citrate buffer. Whenever we are using the DNA probe, one has to use very stringent conditions to avoid any false recognition of the signal. If it is not done, then a wrong interpretation of the results will be there. That is, one will be identifying wrongly any gene of interest from the sample. So this is very essential that gene probe is repeatedly checked whether the reaction is specific or not under the experimental conditions. After hybridization is over, in the next step is the visualization of the gene of interest on the X-ray film by autoradiography. So this is a well proven procedure and it itself is a complete independent technique. So this is not covered in much detail here, but it should be clear that one can easily see the band pattern on the X-ray film after the autoradiography. As far as the applications of the southern blotting is concerned, it is used for identification of specific DNA in a DNA sample. It can also be used for homology-based cloning on the basis of amino acid sequence. Specific probes are designed based on the sequence of amino acid. The probe is further used for screening clones from a DNA library or other collections of the cloned DNA fragments. Southern blotting can also be used to identify methylated sites in particular gene. I must tell that during the 1980s and 1990s, the technique of southern blotting was very widely used in all molecular biology labs, biotechnology labs, and even the biochemistry or any branch of life sciences. But with the development of the newer molecular biology methods, which includes the real-time PCR, the technique of southern blotting is not now much used, but it has a historical significance when the other techniques were not available, and this was the only technique which can be applied for identifying the gene of interest from a given DNA sample. Dear students, let me summarize the topic of discussion. The technique is performed in multiple steps, and you should be careful in every step for successful blot. Common problem is either poor transfer or no transfer of DNA from gel to membrane. Probes can also lead to non-specific detection. Therefore, probe is required to be used under appropriate reaction conditions, which gives signal only in presence of target DNA. The technique was consistently used during 1980s and 1990s, but it has lost its charm because of better techniques available for gene identification. Real-time polymerase reaction is being used for the same purpose for which Southern invented this technique. Nevertheless, the technique was widely used during initial development in biotechnology. At the end, I must thank Dr. Dheeraj Kumar Nanda for assisting me in development of this module.